So picking right back up where we left off and looking at, in this case, let's say these are MRI pictures of different slices of the brain where we can visualize how much damage an individual patient had. And so what you see here are seven it's one orientation, all horizontal slices, but you can see that there are 7 points that the slices come from. And so here this slice orients to that, and so on and so forth. So there's 2, 3 to 3, 4 to 4, all the way to the 7th slice. And then if we look at, for example, this is patient 1, patient 2, patient 3, patient 4, patient 5. And then we can see for patient 1, we can look at all of their slices of their brain and then compare across the different patients and say, okay, like patient 4 and 5 both had damage in a particular area here, and you can look at, patient 1, the damage is visible at slice 3, right? And 4 and 5, and that's the extent of the damage. We don't see at slice 6 or slice 2, we don't see any of the damage. And so it's sort of confined between these different slices. And then we can kind of look at that for each individual person and say, where did the damage begin and end in terms of the location of the brain? And because in an MRI you have 3 orientations, you can really navigate this really well. Now, let's say you have all these individual patients, but then you want to kind of average them together or overlay the different damage together. Well, you can do that as well. So you can have these 7 different slices, and then you can actually overlay based off of, let's say this is the 5 patients. You could say, okay, if all of the patients overlap their damage to the same area, that's going to be colored red. So in this case, in slice 5, you can actually see the overlap here, and all of the patients had the same overlap. Here, all of the patients had the same overlap. Here, all. And then if you look at yellow, that represents this percentage. This represents 50%, so half of the patients had overlap in this particular area here. Or here, the more orange color, whereas more patients had overlap in the yellow. And then now you can start to say, okay, well, let's correlate what these individual patients, their overlapped damage, and say everybody that has damage here, if all of them had issues with, for example, moving their right arm, or sensing something in particular, having a language issue, depending on where, or had a balance issue, depending on where that damage was, then we can say, well, we're pretty confident that that particular damaged area is responsible for this functional behavior. Or that perhaps this particular area of the brain, okay, let's say right here, is connected to somewhere else in the brain that's responsible for that thing. So either we have seen a damage to the pathway or damage to the area where that particular function is sort of housed, or localized is a better way to put it. And then we can also take animals and develop hypotheses about how particular areas of the brain work and function, and then we can manipulate an animal's brain and then to test that hypothesis. So Carl Lashley in the 1920s first started doing ablations, which is basically removing or destroying different areas of an animal's brain, for example, a rat or a mouse, and you can hold them in this device and perform surgery on them. And, ethically, this is something that, some people do argue that, we shouldn't do this at any point. But the idea is that this is translational research. And so we can look at a mouse brain or a rat brain, and we can lesion that brain and say, okay, well, this particular area, when we damage it, causes this dysfunction, and then translate that to humans, because it would be completely unethical to do this on a human, right? But the way that we do this in terms of functional techniques, you can do this several different ways. So you can do a surgical lesion, which is basically where you go in with a scalpel and then you will, slice into the brain and say, okay, well, if you want to go subcortical, then you have to get into that subcortical region. So you're going to have to slice through some of the cortex depending on where you're going. And the problem with that is you're going to damage the tissue. You're going to damage neighboring axons and tissue that might be connected and important for that. And so it might be that behavioral effect is a result from a change that was upstream or downstream from an area, because you severed those connections. Then now you don't know if that is actually the area of the brain responsible or if it's just connected to another area. So what it does is it leads to ambiguity. It's an ambiguous interpretation, and that's really difficult to understand if you are actually getting at the right source. Another thing is that we do have, we compensate. So animals will compensate for a problem if there is, for example, the amygdala. You have bilateral, you have two amygdala, one on each hemisphere, and that's involved in fear response and emotion. And if you were to lesion out one of the amygdala, then the other amygdala would then compensate for that. And so it's really difficult to understand what's doing what unless you take out bilateral. If you have bilateral structures, you take them both out. But there are also other places that we compensate, and animals compensate for injury. So keeping that in mind. So surgical lesions can be really messy. They can be ambiguous in terms of interpretation. We can also do neurochemical lesions. So basically you inject a neurotoxin, and this is better because you don't, especially if you want to go subcortical, you might not be damaging any of the neighboring tissue. So it does help you become more specific. Also, if you want to only target like cell bodies or axons, you can get much more specific with a chemical than you can in surgery. And then one of the better ways to look at functional ability is to temporarily lesion. So you can pharmacologically temporarily lesion something. So essentially you have a chemical that you use, or a toxin that you use that is not a permanent one, and that can you can test the animal prior to the temporary or reversible lesion, and then you would then test them after you've lesioned them, and then once they are recovered from that lesion and you can reverse it, then you test them again, and you can actually do comparisons within the animal itself. Another thing that you can do is cool the brain. Again, this is temporary because that particular area of the brain will then be warmed up and be able to be usable and not damaged, because it didn't need the inputs in at that direct time. So there are some strategies that you can use to examine that functional behavior. And then, like I said, some are better than others in terms of permanence or ambiguity. Other techniques that you can use, so we have PET, right? Positron emission tomography, and functional magnetic resonant imaging. So PET, positron emission tomography, this is a good technique if you want really to see certain aspects of the brain and other parts of the body. It is really helpful to understand metabolism. And it's really helpful to understand certain neurochemicals. Now, both of these, both PET and fMRI, operate on the same principle and theory that the brain needs oxygen to function. And so anytime that you are using the brain more so a portion of the brain more so than the other, you're using all your brain all the time. But there are times when you maybe using something, a structure more so. The idea is that you need more blood flow to that area because it needs more oxygen, because that's how the brain gets its energy. We don't have fat stores in the brain. And so both PET and fMRI use this theory, this idea of blood flow, to look at neural activity indirectly. And so timing wise, it's not great, because we have to estimate how much time you have to process and then get blood to that area. So there's a little bit of a time issue there. But generally speaking, when you look at the pictures that you get, they're much better, and you can see the spatial resolution much better in terms of where in the brain you're looking. So PET scans, like I said, it is important to consider what kinds of techniques that we're using and how dangerous they are. Are they invasive? Are they not invasive? The PET scan is one where we have to use in moderation because we actually do use radioactive chemicals. And so if you medically need a PET scan for a particular reason, let's say their physicians are looking for a tumor, or they need to see glucose metabolism because they suspect you might have Alzheimer's or dementia, you would get this scan for medical reasons. But for research, we have to really limit how many times that we scan people because it is cumulative. So what that means is that you have these radioactive chemicals injected in you, and if you do it too many times, that can make you ill. So you definitely don't want to do that too many times. I'm not as interested for you to know exactly how all of these things work. I just want you to kind of be familiar what you can do with them and what are the limitations of them. And so the PET scans looking at these radio tracers, like I said, you can look at glucose metabolism, you can look at different neurotransmitters, depending on what you're targeting. So it's a really interesting technique to use if you're interested in some kind of metabolism. Here you can see a typical brain. Here you can see a brain with Alzheimer's disease and one with frontotemporal dementia. And so one of the other theories of dementia, and especially Alzheimer's, is that it could be a glucose metabolism issue. Because we do see the ability of a person with Alzheimer's to not be able to metabolize the glucose as well, especially in the in certain areas of the brain. And so we definitely can keep this in mind. We'll talk about Alzheimer's later on in the course, and we'll talk about it actually later on in the slides. But PET could be a really good way to visualize this because as you can see how the processing looks different based off of the color coordination is different from each of these brains. So it's a really good technique to visualize metabolism in the brain. But again, we have to be careful about how often we do this with our participants. Another thing, thinking about fMRI. So fMRI is functional MRI, uses an MRI machine. You put a person in the scanner and have them do a task. Same thing you can do with other techniques that we'll talk about later. But this is one that is based on the idea that we need blood flow to a particular area. So it's looking at the amount of oxygenated versus non-oxygenated or deoxygenated blood. And so it's this bold signal. It's blood oxygen level dependent. So again, the idea is that when you use a particular portion of your brain more so than others, then it will this analysis that you do. So if you're in the scanner and you're looking at a picture, or you're watching a video, or you're listening to a song, that those areas of the brain that use more during those tasks will be in need of more blood. And that is what fMRI is looking at. It's looking at that blood flow and the hemoglobin, oxygenated versus deoxygenated blood. And we get these really nice pictures, right? So you can say, okay, the areas where they're brightly colored, the really bright like white is the areas that are getting the most blood flow. And then all the peripheral areas are not getting as much. And this is subtracted from the normal blood flow that you would see in the brain. So this is just concentrated to just the brightest areas. And so you might say, okay, when you are listening to some kind of sound bite, or you're listening to someone speak, you would expect your temporal lobes to quote unquote light up. And that's exactly what we see here. Is we anticipate that this person's listening to something, and the extra blood flow is going to the temporal regions because that is where we are using more of our neurons. However, the one bad thing about fMRI is it really only considers turning things on, right? Neurons that are working. And sometimes it's just as important to consider the things that aren't working, because inhibition is also an important step. It's not just excitation. So sometimes for something to work, other things need to be turned off. And that's something that we're going to talk about later when we get to neurochemistry and when we get to action potentials and stuff like that. And then finally, the last one I want to cover is just about this optical imaging. So the same kind of idea, looking at the blood, the oxygen level that's in blood, and bouncing light off of the cortex. And so this is a cap that you would wear. It's non-invasive. You just place this on top of your scalp. And basically it shoots these sort of rays of light. And so one downside is it's only measuring this part of the cortex. It's just getting at the very outer layer of the cortex. And it's just measuring the sort of oxygenated, deoxygenated blood there, the hemoglobin there. And, it's got to get through the scalp. It's got to get through the skull. And it only takes this sort of portion of the cortex. So it's a really interesting technique. So they call it NIRS, near infrared light spectrum, or spectrography. And this optical imaging is interesting. It's not used very heavily because it's rather limited. It can only do those outer layers of the cortex. If you wanted to go subcortical, you would have to use fMRI. You would have to use different techniques because this is just looking at that sort of outer layer of the cortex.